to chapter 15 in our biochemistry series of chapters. This is going to be talking about lipids. So we've looked at carbohydrates or sugars. Lipids you may be familiar with when we talk about fats or fat stores, but also you might have heard like cell membranes or like what we call the lipid bilayer. That's what we're going to be talking about here in lipids. So what we're going to be talking about first, an overview, is we're going to be talking about something called fatty acids. So fatty acids are the basis for what we talk about with lipids. So you may remember this a little bit from your organic chemistry chapters, but fatty acids, by definition, the name, we have an acid portion or a carboxylate, so which is the acid portion, a carboxylic acid, followed by an R group, or what we call a hydrocarbon tail. So again, in this case, you have, if we draw our hydrocarbon tail on our skeletal structure here, you have one portion of the fatty acid, which is nonpolar, and you can have another portion of the fatty acid here, which is polar because of the oxygens that are present as a result. So we will go through how we look at the nomenclature, naming, and drawing of fatty acids. Then we will put those fatty acids together to make something called triacylglycerides or triacylglycerols. So tri for three. It's actually what we're doing is we're putting 3 fatty acids on a 3-carbon compound called glycerol. From there we will talk about membrane lipids. So these are the ones that we talk about when we talk about those lipid bilayers. We have phospholipids that contain a phosphate group. And we have glycolipids. If you remember from our polysaccharides portion, when you put glyco in front of something, it means sugars. So you can put a sugar on a lipid. Remember we talked about how sugars can be used for signaling molecules? Well, the lipid bilayer on the outside of cells can have a signaling molecule then with a sugar that goes out to the outside world to, again, transmit those communication signals. And then finally, we will talk about the other components of lipid bilayers, proteins, for example, or what we call lipoproteins. So chapter 16 talks about proteins, which will be our next in our 4 chapters. But we also talk about cholesterol. Now, normally we've seen cholesterol as a bad thing, and cholesterol is one of those components where too much is a bad thing, but also too little is a bad thing. So you just need the right amount. So we will look at those. Okay, so if we take a look at lipids by definition, so lipids are actually the most diverse of all of the 4 major macromolecules. So again, just to reiterate, our 4 major macromolecules that we will be looking at in this class are, again, carbohydrates or sugars. That's what we looked at in chapter 14. We are going to look at lipids or fatty acids in this particular chapter. We are going to look at proteins in the next chapter, or amino acids that make up proteins. And then finally, we will talk about nucleotides in our final chapter. So these are our 4 major macromolecules. So remember in chapter 14, when we talked about carbohydrates, we talked about glycosidic linkages. Alpha, right, opposite, beta, same. So we have linkages between to make disaccharides and polysaccharides, etc. In proteins, we get a protein by making covalent bonds between amino acids. In nucleotides, we get strings of DNA and RNA by attaching our nucleotides together. Lipids are a little bit different. Lipids actually, there's no covalent linkages between the individual fatty acids to make the molecule, but instead what these lipids tend to do is they tend to aggregate. And what this actually has to do with is it has to do with the fact that things that are polar want to be near other things that are polar, and things that are nonpolar tend to be near other things that are nonpolar. So if you've ever seen sort of a cartoon description of a lipid bilayer, what you might see is we might see these little circles as our polar, what we call polar head groups. And those are going to face water because water is also polar, unlike the outside of the cell. And then you might see like 2 little squiggles. And these are the hydrocarbon tails that go down. So here we have our nonpolar groups. So in this case, what you see is the individual lipids are not actually physically bonded together, but they come together based on those properties in terms of polar species and nonpolar species, so they what we call aggregate together. So they actually don't form these polymers, right? A protein is a polymer of amino acids. A polysaccharide is a polymer of individual amino acids. A polymer of DNA and RNA is made up of individual nucleotides. We don't see that with lipids. So we are going to talk specifically about how do we get the architecture of what these lipid bilayers look like, but also how do we also use lipids and fatty acids for energy stores as a result. So this is a little diagram that you can see in the slides here to the left of what we're going to talk about. It's a nice kind of flow chart that I kind of like to help describe how we put these pieces together instead of just describing them individually. So we are going to start with fatty acids. So if you notice when we go to fatty acids, we are going to talk about what we call glycerol-based lipids. We'll talk about sphingosine-based lipids, and then we will talk about, on the far right-hand side here, these other components of lipid membranes like cholesterol and steroid hormones and things like that. So let's first talk about these fatty acids. Again, you may remember them a little bit from your organic chemistry chapters, but this just shows you some examples in the book. But there's basically 3 different characteristics that we need to look at to describe a fatty acid. The first thing that we need to describe is similar to carbohydrates. Well, how many carbons are there? Now, the nice thing about sugars is we could have things like 3, 4, 5, 6. That was usually the limit to the amount of carbons that we would see in a single sugar, in a monomer. In hydrocarbons, we tend to see usually, the hydrocarbon chain in a lipid, it's usually 12 to 20 carbons that we have there. So you need to specify how many carbons does this particular fatty acid have. Secondly, you need to ask, okay, are there any double bonds? So if you remember back to your organic chemistry chapter, right? If you have a double bond, it's an alkene. If you have a single bond, it's an alkane. So you need to say, hey, how many double bonds do I have? If you have 0 double bonds, you are what we call a saturated fatty acid. So you may have heard, or if you look on the back of certain labels of things that you may buy, you may have heard of saturated fats or unsaturated fats. That literally has to deal with, hey, in these particular fatty acids that I have with those hydrocarbon tails, do I have double bonds, yes or no? That's all that's referring to, okay? So if you do have double bonds, so if you have 0 double bonds, you're saturated. But if you do have double bonds, if they are present, then you are therefore unsaturated. So if you think about it, we're thinking about saturated here. Saturated means that you're basically full, or you're saturated, of hydrogens. So again, because if you put in a double bond, you decrease the amount of potential hydrogens that you can have on that backbone. So a double bond means you're unsaturated. So you may have heard of things like monounsaturated, mono meaning 1. Monounsaturated means there's 1 double bond. Polyunsaturated, more than 1 double bond, those types of things. So however, here's the deal. So you need to specify, well, how many carbons do I have? How many double bonds do I have? But then you also need to say, where are my double bonds? So there is information also within this. You can also think about what type of double bonds, and this has to do with something called the isomers of cis versus trans. So maybe in the news and things like this, you may have heard of people saying, oh, trans fats, and trans fats are bad for you and things like this. Well, trans fats have to do with basically how the positioning of the fatty acid goes. So if you can look at this is from your book, just showing you this. So you can review chapter 12 for some of the background if you're kind of unsure. That will kind of give you a good refresher. But if you take a look here with part B with oleic acid, it actually has a double bond that is cis. So those hydrogens are on the same side, okay? So you get kind of a kink that goes down. But when you have a trans double bond, it keeps it nice, it keeps it more straight because the hydrogens are on opposite sides of that double bond. So again, that's what we're kind of looking for when we look for the overall characteristics of a fatty acid. We need to specify how many carbons does it have, how many double bonds does it have, and where, and the type of double bonds that we have. So we're not necessarily going to look too much at the type of double bonds because most of the double bonds we see in us are cis. There's occasionally some trans, but that's about it. We're going to look more at the nomenclature to specify the locations of those double bonds. And here's the thing. If you take a look at these characteristics are really important because these characteristics lead to differences in physical properties, such as, if you see in the slides here, melting point or boiling points, things like this. So, for example, I'd like you to think about butter versus like olive oil versus coconut oil versus avocado oil. You've seen all these different oils around. Butter is a saturated fat. So at room temperature, butter is usually a solid, okay? But something like olive oil tends to be unsaturated. And depending upon how many double bonds you have and where those double bonds are located and the stereochemistry of those double bonds, you can get different boiling points and melting points that you see, and you'll see some oils that will melt at different temperatures, okay? So that's why it's important for us to get this naming down, because that naming helps us to figure out the physical properties of these fatty acids that will be very important. So how you do this. So again, we have these 3 particular species, that characteristics that we need to look at. 1 and 2 here are actually pretty straightforward with how we do naming. Number 3 is the one that is a little more difficult. So number 1, what you do when you're naming a fatty acid, you put the number of carbons, you put a colon, you put the number of double bonds, and then after the number of double bonds, you have 2 ways of determining the position. One example is we put a delta as a superscript followed by a number, and the number of these that you have here matches the number here for the number of double bonds. Okay, that's the more common way that we use this. So for example, if we take a look at the example in the slides here, you have palmitic acid as an example. So palmitic acid by definition has 16 carbons and no double bonds. So you would not have to specify the positions of those double bonds because you have none. So we would just put 16, colon, 0. Boom. That would be it. So you notice for those saturated fatty acids, pretty straightforward of how we name them. However, when we have double bonds, we have to specify again, how many do we have and where they're located. So if you look at the example here in your slides, you can see we have something called linoleic acid. Linoleic acid has 18 carbons. It has 2 double bonds. So that means I need to specify at what positions do these double bonds reside. So you do this by taking a delta. So a delta means a change. So you want to figure out where does that change from saturation occur? That's essentially what you're looking at. So if you name the carbons that you have, so in linoleic acid, you again have carbon number 1 because that's your highest priority functional group with that carboxylic acid. Carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons. Now, if you look at that nomenclature, we have a delta, so there's a change at carbon number 9 and carbon number 12. So we put 9, 12. So if you notice, there's a 2 here, there should be 1, 2 numbers here. If this was a 3, there should be 1, 2, 3 numbers. If this was a 1, there will be 1 number. Okay? So again, the number that you have after the colon here, if you use the delta nomenclature, you should have 3, or you should have the same number of species here that equal this. Okay? So now we know there's a difference at carbon number 9 and carbon number 12. So we start counting from carbon number 1 here when we use the delta nomenclature. So we go 1, 2, 3, 4, 5, 6, 7, 8, 9, and then we draw our double bond. Again, I am not interested necessarily in you guys understanding the cis versus trans. I'm just looking at getting the positions of where those double bonds are. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and then you draw the double bond between 9, 10, 11, 12. And we draw our second double bond. Okay? So you may have heard of omega-3 fatty acids. So if you notice, the difference between 9 and 12 is 3. So a lot of these omega-3 fatty acids will have numbers that differ by 3. Okay? The other way that you can do, so that's the first way of doing nomenclature for fatty acids. So please make sure you're getting extra practice using the homework and the questions in the back of the book. You should be able to, if you're given this nomenclature, draw an example. Or if you're given the drawing, you should be able to give this numerical nomenclature. So the other way that we do this is we can also specify, because that omega-3 that I talked about is a difference of 3. That's a very common trend in fatty acids. So what you can do is, if you know that it's called an omega-3, if you remember in the Greek alphabet, omega is the last letter or the last carbon. So the other way that you could also write this nomenclature would be 18, 2, and then you in parentheses would put omega minus 6. And what this means is, rather than starting from carbon number 1, you start from the last carbon, count back 6, and then the double bonds are always in positions where they are 1, 2, 3. So there should be 2 single bonds in between the double bonds. Okay? So these are separated by 2 single bonds. So if we were to draw this, we would actually draw the exact same thing. However, I'm going to get rid of these double bonds because we can draw 18 carbons, just like we had. But now, instead of starting from carbon number 1 and counting like we did with the omega, or with the delta, excuse me, we're going to start with the last carbon here. So we will count from here 1, 2, 3, 4, 5, 6. And then we will draw a double bond. And then you will count double, single, single, and then the next double bond will go here. Now, if this was, instead, let's say this was an 18,3 omega minus 6, our next double bond would go double, we would have a single, and then another double bond. If it was 4, we would again go single, single, double, etc., etc. So that's always the same pattern. So you should know both of these ways of looking at the nomenclature for naming these fatty acids. Okay? So again, here are some examples. This is just from your book, but it shows you, for example, some of the common, okay, unsaturated and saturated fatty acids. So myristic acid, palmitic acid, 14,0, 16,0. Could you go ahead and draw those, right? Just as we've drawn here with these skeletal structures. Could you also draw, for example, oleic acid, which is an 18,1, okay? And then you should also, based on the condensed formula that they give here, could you put the nomenclature for what the delta would be and for what the omega would be, okay? So if you take a look, we just did linoleic acid. So if you notice here, you've got, right, your number of 6, 7, 8, and this would be carbon 9. So 9 to 10 has a particular double bond. And so looking at that, and then you do that again twice, so that gives you a 9 and a 12 is the location of where those double bonds are. So if it helps, you can, as you're drawing out your fatty acid, you can label your carbon numbers, right, to help orient you as to where this would be. So if you notice here, we have a new double bond at carbon number 6. So this would have the nomenclature 18,3 delta 6, 9, 12. Again, 3 numbers here, there has to be a 3 here. Those numbers must always match. So why is this important? Well, again, just like we talked about with butter and olive oil, right? The number of double bonds and the number of carbons that you have change the physical properties of these species. So if you notice, look at those melting points. When you look at those melting points, the more, as you increase the number of carbons, so going from myristic acid to palmitic acid to oleic acid, or stearic acid, excuse me, you have melting points that increase. So as we increase numbers of carbons, our melting points go up. They're more stable. Okay? It takes more energy to melt them. But when you start introducing double bonds, what do you notice happens to your melting points? So here, if you look on the slides, 18,1, 18,2, and 18,3, our melting points get lower and lower and lower. So again, as we increase the number of carbons, we increase our melting point. As we increase the number of double bonds, we decrease the melting point because there's more fluidity in there. So again, the length of the number of carbons that we have, the length of this hydrocarbon tail, helps us to determine the physical properties, but also the saturation level of those fatty acids help us to determine those physical properties as well. So from here, the next thing that we're going to talk about now that we understand how fatty acids are put together are talking about something called triacylglycerides.